Okay, so we're going to end the semester talking about tumor suppressor genes. So this has been a three-part series on cancer. At the beginning, we talked about just kind of overview of the behavior of cancer cells, a little bit of the history of some of the early discoveries that led us towards an understanding that changes in genes and genetics gives rise to cancer. It could be through random mutations, or it could be through carcinogens or mutagens that we encounter in the environment. And then talked about oncogenes and how they were discovered and what they do and their role in cancer. And generally, the oncogenes become activated through mutation, and they drive cell division, or they drive metastasis. It's considered gain of function. They're doing things that they shouldn't be doing. Now, when we talk about tumor suppressors, actually it's the opposite. That is, the tumor suppressors are normal important genes in the cell, and their role is to inhibit cell division under various conditions. In response to DNA damage, the tumor suppressor genes like p53, there's lots of others though, will inhibit cell division, during which time the cells may repair the DNA, or they'll induce apoptosis to eliminate severely damaged cells. There's a whole series of genes that their role is to inhibit cell division under particular circumstances. Now, when these genes are defective, then the cells lose their ability to inhibit cell division under those circumstances. So what we see with the tumor suppressor genes is they're, as I said, as I've written here, they're characterized by a loss of function. That is, they can be deleted completely. Or there can be major mutations that eliminate the gene. You can lose a whole chromosome. Often there are deletions that remove parts of the gene. In any case, they become non-functional. So the loss of the tumor suppressor gene activity leads to uncontrolled cell division. The activation of the oncogenes leads to uncontrolled cell division. So it's two different, in a sense, two different pathways. Now, in many cases, the tumor suppressor genes are associated with inherited cancer syndromes. That is, syndromes that you see inherited among families. You don't inherit the cancer, but you inherit the predisposition, the likelihood of getting the cancer. And I'll explain that in a few minutes, but I'll just briefly say that the reason that is because we have two copies of every gene, of course. Okay, sorry about that. That was a phone call I picked up. Anyhow, so where was I? I think, okay, yeah, inherited cancer syndrome. So I'll just briefly tell you, I'm going to talk about it, give you an example, a specific example in a few minutes. But they are, the tumor suppressor genes, the loss of function is associated with these inherited cancer syndromes, where you have, in a family, people inherit a predisposition, the likelihood of getting cancer, like, for example, in inherited breast and ovarian cancer with the BRCA1 gene, which is a tumor suppressor gene. And the reason is because we have two copies of each gene, right? We're diploid, two copies of each gene. And if you are born with a defect in one of the two, then the second normal one generally will suffice, so that that individual will be born and will be normal, except they are predisposed for the loss of the second allele in a given cell type. So once you lose that second allele, you've lost both tumor suppressor genes, or both copies of the tumor suppressor gene, and you get cancer. So it can be inherited. You can't inherit the the activated oncogenes, because they're so dramatic with their activity that usually it wouldn't be viable. But you can survive quite well with one copy, and then you lose the second copy over time. Okay, so let's take a look at some more details. So as I mentioned, the tumor suppressor genes play a role in inhibiting cell division. Sometimes they're important for inducing apoptosis, which is a programmed cell death. But either way, you get a loss of cell division. Either the cells stop dividing, or they disappear. And they're characterized by being inactivated or lost or deleted in many tumor cells. And here I just gave some examples. There's a number of these that have been identified. And so I'm going to list some of these out. Lee-Fraumeni syndrome is a disease, an inherited cancer syndrome, which is due to loss of p53. There's Wilms tumor, which is a kidney tumor, a deletion or loss of WT1, colon cancer. Another one, loss of a gene called APC. Breast and ovarian cancer, the BRCA1 gene probably most of us have heard of. Neurofibromatosis, gene called NF1, and retinoblastoma is a tumor of the retina, which is due to deletions or mutations, inactivating mutations in a gene called RB. Now, what's characteristic of these tumor suppressor genes is that in order for the tumor to develop, both alleles of the gene need to be lost. We have two. One we get from the mother, one we get from the father, so everything is in twos. And in order to get the cancer to develop, both alleles need to be lost. And that gives rise to the finding that these can be inherited. There can be an inherited syndrome, because you can inherit a mutant allele from one parent and inherit a normal allele from the other parent. And so you have one normal or wild type and one inactivated, and in many cases, the one normal copy is sufficient for the individual to have a normal phenotype, no cancer. But since every cell in the body has only one copy, and all you need to do is lose the second one, then there's a very high probability that will happen and you'll get cancer. And that is the basis for the inheritance. So all the cancer, all the diseases that I have listed on the left-hand side can express themselves in an inherited predisposition. Lee-Fraumeni, Wilms tumor, colon cancers can be an inherited predisposition. Breast and ovarian, we know. Neurofibromatosis and retinoblastoma are all considered inherited cancer syndromes. In the next slide, we'll just kind of talk a little bit more why that is. So with all these tumor suppressor genes, the cancers that arise from the loss of these genes actually kind of occurs in two forms, two presentations. One is sporadic, that is randomly in the population with no family history. It just sporadically comes up, and the other is the inherited predisposition. Now let's first of all take a look at the inherited predisposition, which is shown on the right-hand side. So there's a number of characteristics that take place when there's an inherited predisposition to a cancer. The first is that people usually get these cancers at very young age. Children could be 3, 4, 5, 6 years of age. Generally speaking, when a child gets a cancer, it's usually associated with some kind of a inherited predisposition. The other thing that we see with retinoblastoma is that first it occurs in children, and they get tumors in both eyes, and they get multiple tumors. So it's a very aggressive disease. Multiple tumors in both eyes. They get it when they're very young. If they are cured of the tumor, of these retinal tumors, then later on in life they often get another cancer, which is bone cancer, osteosarcoma. So these people are destined to get some kind of cancer. So it's very frequent. If you look at the sporadic case, it's extremely rare. It happens, but it's extremely rare. The estimate is about 1 in every 30,000 people will get retinoblastoma. When they get it, it's usually as an adult, and it's much less severe. They get one tumor, which can be treated. So the pedigree, this small pedigree below, shows an inheritance of this. So the father on the top has a wild type, which is the plus, and a mutant retinoblastoma gene. The mother has 2 copies of the normal, and then it goes through normal inheritance. And so what's showing here is that the male, the male offspring, is the one that inherited the mutant retinoblastoma gene. Now this is the child who will get tumors in both eyes, multiple tumors, and later on osteosarcoma, likely. The female is completely normal, and she will not show any symptoms. So if we look at that male, the male actually is normal. He's a heterozygote. He has one normal copy of the retinoblastoma gene, and so early on, early in life, I mean, for the first few years, everything is fine. But because there is a loss, the cells are a little bit weakened, and there's a tendency to have a mutation occur or some kind of defect in that second allele. And because there is some kind of instability because of the function that the retinoblastoma gene has in the cell. But more importantly is, all the cells are predisposed. So you have estimated 6 million retinal stem cells per eye. 6 million. We have 2 eyes, so that's 12 million cells. All of these are heterozygotes. So all you have to do is lose that second wild normal allele in one cell, any one of those 12 million, and you get the tumor. And so it's not an infrequent event because the cells are a little bit sensitized because of they are heterozygotes. They only have one copy, so they have less of the protein around. And so there is a very high likelihood, or maybe it's not even that high, but there's a likelihood that there will be a loss of this normal allele, and then that person would get tumors. So if the child has 2 tumors in both eyes, that means it happened in 4 different cells, which is 4 out of 12 million, which actually is a low probability, but still it happens, you get the tumor. Now there's there's so many cells, and there's a likelihood, a fairly high likelihood that one of those 12 million cells will get the deletion or the mutation in the second allele and get the tumor, that it's considered 100% penetrance, meaning that every child born as a heterozygote will, over the course of the next couple of years, few years, maybe 5 years, will end up getting a tumor. So if you were going to set up a pedigree, you would see that that person getting the tumor, it looks like an autosomal, it inherits like an autosomal dominant. The father would have had it, maybe he was cured of it. So it inherits at an autosomal dominant. So that's why we see it in the inherited form, occurring young, both eyes, multiple tumors, and then also other cancers later in life. The sporadic form, it's just very rare because the allele both alleles have to be lost in a single cell. Whereas in the inherited form, they've already lost, every single cell in the body has already lost one. In the sporadic form, both have to be lost in the same cell, and that's why you see it at a much lower frequency, 1 in 30,000. Takes a long time to happen. If anybody gets it, the person's an adult, and they usually only get one tumor. And this is the classic inheritance pattern that you see with all of the tumor suppressor genes. So both sporadic and inherited forms. Okay, let's go on to the next slide and kind of look at see what these tumor suppressor genes are doing. And we'll use p53 as a model. So p53 is a classic tumor suppressor gene, as is retinoblastoma. P53 is probably one of the most well-studied of the tumor suppressor genes. It is involved in an inherited cancer syndrome, that's Li-Fraumeni syndrome, which I'll come back and talk about in a moment. It's also involved in just about every single cancer that we're aware of. It is not tissue specific like retinoblastoma, just retinal cells, osteosarcoma. The APC gene is involved in colon cancer. The BRCA1 is involved in breast and ovarian cancer. But the p53 gene seems to be so central to so many different processes that defects in this gene can give rise to cancer in any cell type. So we see it in brain, we see it in lung, we see it in kidney, we see it in skin, we see it in ovarian, colon also. It's just whatever tumor you look at, p53 seems to be involved. So what does this gene do? In normal cells, p53 functions as a DNA binding transcription factor. That is like what we talked about, the genes that get activated. I mentioned p53 at that time. p53 binds to a specific DNA sequence upstream of many genes and activates its expression. Activates their expression. Actually, a few different classes of genes. And in general, it is turned on in response to DNA damage. And that's what's shown in the middle section. So in response to DNA damage, this could be damage from UV light, it could be damage from X-rays, it could be damage from compounds that could damage DNA like the bulky adducts, all these kind of things. What happens is p53 gets activated. The protein becomes activated by the addition of a phosphate group or phosphorylation. And then it can induce a number of different pathways. It can induce DNA repair genes. It can induce G1 arrest, so the cells stop dividing. Presumably, what happens during that period is that the DNA repair genes that have been activated repair the DNA. It can undergo G2 arrest, depending on when the DNA damage occurs. It can block DNA replication, and it can also induce apoptosis if the damage is so severe. And in some circles, it's referred to as the guardian of the genome. That this is the most important, one of the most important proteins or genes that help stabilize the DNA in response to all kinds of damage and instabilities. As I mentioned, it's mutated or defective in greater than 70% of all cancers. So you get all different cancer types. Now, if you look back at the original list that I showed you of the different genes that were activated, that were inactivated in tumor cells, list of some of the tumor suppressor genes, p53 was the first one up there, and the disease that I associated with it was Li-Fraumeni syndrome. Li-Fraumeni syndrome is an inherited cancer syndrome, analogous to retinoblastoma. That is, individuals with Li-Fraumeni syndrome get tumors at a very young age, 6, 8, 10 years old. But because in the sporadic form, mutations, defects in p53 lead to all kinds of different cancers, like I said, skin, lung, kidney, brain, all different kinds of cancers. You see the same pattern in Li-Fraumeni syndrome. Whereas with the retinoblastoma, they get the retinal tumors. With Li-Fraumeni syndrome, they can get all kinds of tumors. They can get, these are children, bone tumors, soft tissue tumors, brain tumors. It reflects the same pattern as it does with the sporadic, only that it's an inherited syndrome. That means they are born with one mutated copy of p53. This weakens the cell response to DNA damage, and so there's an increased likelihood that other genes will be damaged, and p53 gets damaged. There's 2 down, and then you get the tumor. So that's kind of the p53 role as a tumor suppressor. Let's look at the next slide. Now, as it turns out, since p53 is involved in the response to DNA damage or other kinds of severe stresses to the cell, and the fact that it is mutated or defective in such a high proportion of human tumors, it has become clearer over the years, and people looking back at the data of how people respond to chemotherapy, that mutations, defects in p53 cause patients to have a much poorer response to treatment. And that, in part, is because many of the treatments that are used are chemotherapy agents, like the alkylating agents, or radiation, which causes damage to DNA. And in many cases, when these cancer cells are treated with these agents, the reason why they're killed by these chemotherapeutic agents is due to their ability to undergo apoptosis. So when they're treated with these agents, the cells die in many cases, not all cases, due to apoptosis. Now, if p53 is defective, which it is in many cancers, then the cells do not undergo apoptosis because they depend on p53 to undergo that pathway. So the finding is that in many cases, tumors that have mutations in p53 are much harder to treat because they do not undergo the apoptotic response, which is the way that numerous chemotherapeutic agents eliminate the cells. If the drugs are used at high enough levels and the radiation is used at high enough levels, then there's so much damage to the DNA that the cells institute other pathways and end up eventually dying. If you break the DNA into multiple pieces by using X-rays, then the cells may go through another round or two of DNA replication, but they can't sustain it just because the DNA is so damaged. But they're not undergoing the apoptotic pathway, they're undergoing some kind of senescence or just basically cell death. So p53 also induces the apoptosis, which is required for many chemotherapies. And if you look at this chart here, what this is showing is the percent of cells dying in response to X-ray dose. And what you have at the top is 100% cells viable, and at the bottom, on the left-hand side, 100% cells viable. And then as you drop down, there would be 0% cells viable. And so what you see is if you have cells that have wild-type p53, normal cells, and you treat them with increasing doses of X-rays, what you see is the cells die. Over time, they lose their viability, they undergo apoptosis. The cells with mutant p53, which are shown up at the top, they're hardly affected by even very high doses of X-rays. And generally, the X-ray dose may be between 5 and 10. The term is gray, but it doesn't really matter. And so you can see treating cells that have mutant p53 in a cancer with X-rays may not be very effective. So once we know the genotype of the tumors, it becomes easier to decide on the treatments. So we talked about oncogenes, we talked about tumor suppressor genes, but it gets a little bit more complicated. There are some cancers which are due to defects in one or very few genes. But in many cases, in most cancers, a number of genes need to be defective, both oncogenes and tumor suppressor genes, before you get a full-blown cancer. And this is why, generally speaking, cancers are diseases of older individuals, adults, elderly, that over time mutations occur and they accumulate and in a number of different genes. And so it takes time for these things to happen, which is different than what happens with the inherited cancer syndromes, where you're already predisposed because one or more genes may already be defective. So I want to just show you we're going to close with the classic example of colon cancer, where people have been looking at the genes that have been involved and see that there are numerous genes and it takes some time. So let's look at the last slide. So overall, when we look at cancer and we look at the genetic defects in cancer, it turns out there are many genetic defects, many genes that are affected. And in fact, that's the reason why cancer generally occurs in older individuals, because it takes many, many years to develop all the different mutations that are required to get full-blown cancer. It's a little bit different with some of the tumor suppressor genes and the inherited cancer syndromes, because these individuals are already born with a severe genetic defect, the loss of one of the tumor suppressors, and so quite often with some of the inherited cancer syndromes, you can see children getting the cancer. And if you ever see a child with cancer, it usually points to some inherited predisposition. They're already set up. They're born with this predisposition, so it occurs. The other mutations need to occur can occur much earlier. But the fact of the matter is, it generally takes a long time to develop cancer, many years, and that's because there's many defects and many mutations that need to occur to get a full-blown cancer. And the model that's been used and studied quite extensively is colon cancer. And this represents work that was done at Johns Hopkins, many years looking at colon cancer, led by an individual named Bert Vogelstein, who's been working on colon cancer for many, many years. And really the reason he decided to work on the colon cancer is because you can visualize different stages of the cancer. If you do colonoscopies, you can look and you can see normal epithelium. You can see what's called polyps, which are early adenomas. You can see them growing a little bit bigger. You can see what's called hyperplasia, that is a kind of a overgrowth of some of the cells. So you can actually visualize different stages of the cancer. And so that's what's shown in this diagram here. You start out with normal epithelium, then you get this hypergrowth of the epithelium, kind of like a swelling, small polyps, which would be an early adenoma. Then you get larger polyps, which would be a later adenoma, and then even larger, and they start getting maybe more more damaged. The tissue becomes more damaged, and you get a carcinoma, and eventually you get metastasis. So there's all these various stages. And what he did was he did genetic analysis on all these stages and found, and this was quite routine, he saw this in all of these samples that he looked at. He looked at hundreds and hundreds of colon cancers at various stages of the progression, and routinely found multiple genes that were defective, and those genes became defective and gave rise to the next stage. So at the beginning you have APC, a mutation in APC, which is actually a tumor suppressor, and then you get DNA hypermethylation, so there's some changes in the structure of DNA. Then you get mutations in RAS. Then you get loss of another gene called DCC, which stands for deleted in colorectal carcinoma. Then you get a loss of P53. Then they say there's other alterations that take place, and the cells eventually metastasize. So the bottom line is that it occurs that all cancers occur in multiple stages, and there's many genes involved, loss of tumor suppressors, as well as activation of oncogenes. Now, to also tell you that with respect to the colon cancer is probably, I believe, it's the only cancer that can actually be prevented. And that's because you can do colonoscopies and remove those early adenomas if they're seen, or even intermediate adenomas. And then you eliminate the possibility of them developing further. So if everybody, they recommend having colonoscopies, I think, when you turn around 50, maybe once every 10 years, unless you see polyps, they get taken out, and the risk of cancer disappears. So it's extremely important. Now, the problem I talked earlier about the early onset colon cancer, that's a little bit of a problem because people are getting them at the 35, 40 years of age, way before there's recommendations to get these colonoscopies. And so there's efforts to try and change the ages at which people are recommended to get the colonoscopies, get them earlier. Now, there's also other developments that are going on to check for colon cancer or polyps by other means. Blood tests have been used. Stool samples have been used. But for right now, the colonoscopies are really the right way to go. They're foolproof. You see the polyp, take it out, done. Okay, so that pretty much ends my discussion of tumor suppressor genes, but also wanted you to know that there's multiple genetic events that need to take place to get full-blown cancer. So that is it. The end of the semester has come. For those of you who are planning to take the final, if you go through your grades, add them up, they should all be up there by now. And it's out of 400 points, and basically just calculate whether or not you need to take the final. If you're a little bit confused about how to figure out what you need to get on the final in order to get a better grade, you can contact me. We can do that together. But in any case, have a good rest of the semester and a good summer. Hope things lighten up over the next few months, and when we come back to school next fall, we'll be able to meet in person. All right. Again, if you need any advice about taking the final or not taking the final, which is optional, just send me an email. We can do a Zoom call, or we can talk about it. All right. Take care, everyone.